Yeah, but I still wish I could have done for other things. I would suggest getting someone else if you don't want to do yourself. With all the stress, it's just too much.
All right, today for lecture, we'll start off with the Neuromuscular Junction, still in Chapter 9. Now, like the bone chapter, we started off with a case study. In this chapter 2, the case study has to do with defects at the neuromuscular junction. So the case study for this chapter involves Annie, who's a swimmer. She's on a college swimming scholarship. Recently, she had been feeling tired, and her times have been getting slower. She's also noticed that her vision is often blurred. Concerned, she goes to see her doctor over the midterm break and is referred to a neurologist who finds that Annie is suffering from something more than just fatigue. So in this case study, we'll take students through the series of stages that describes Annie's problems and symptoms. And you guys need to use your knowledge of the neuromuscular junction, nerve and muscle physiology, to fit pieces together and determine what is wrong with Annie. So once we go through the details of this, this condition will be revealed throughout the lecture. So we're going to start off by talking about, there's a picture of the neuromuscular junction right there. It's the interaction between the nerve ending and the muscle membrane. We did mention how calcium is liberated from the triad and how that initiates a cross-bridge formation between actin and myosin. But to take that further, we'll talk about the events called cross-bridge cycling, where muscle actually contracts and shortens. That's kind of an overview for today's lecture. So in this picture from the book, it's kind of an overview where they show a person's muscle in a state of contraction being flexed at this angle. And that's just to hold the device she's looking at. There's a nerve that innervates muscle fibers, and it kind of, we focus in on the interaction between the nerve ending and the muscle fiber there. So you have to consider two cells. What you can't quite see from here is that the beginning of the nerve, or this nerve ending, is actually in a section of the spinal cord, which I always draw like this. Think of the spinal cord in sections. It's a segmented structure. And in each segment, there is a number of spinal nerves that are issued from it. So, for example, there's the cell body, specifically of the nerve that's in this part of the spinal cord. It's called the ventral horn. We'll learn that later. But the cell body, which is the nerve cell, it's in the protection of the spinal cord. The spinal cord is in the vertebral column. And there's a specialized process when this cell is stimulated. It leaves the spinal cord, and it's going to go all the way to a muscle fiber. So if I can draw it bigger. So like this nerve ending, I'm drawing bigger. It's supposed to symbolize this. That's called the axon terminal. The name of this cell that starts in the spinal cord there, it's called a motor neuron. Neuron describes the cell type. It's the major cell of communication in the nervous system, which we'll talk about. Motor is a function. A motor function means you command your muscles to move, in this example. Okay, so this axon terminal, it terminates on a muscle fiber. So a muscle fiber, if that's the cell membrane of the muscle fiber, we call that the sarcolemma. The part where the nerve ending communicates with the sarcolemma, it's kind of like pushed in and it's all like folded and convoluted. And there's all these places where there's like, you'll see it more, they're really small, these little purple channels. I'll try to leave a space for that. I'll draw a few. Well, anyways, this area where this nerve ending interacts with the muscle fiber is called the motor endplate sometimes. We're calling it the neuromuscular junction in this lecture. So this interaction, I didn't draw it very well, but motor endplate or neuromuscular junction. So let me advance the slide. The details of what happened at the neuromuscular junction are shown in this figure here. And they take you through a series of steps where essentially the neuron stimulates the muscle to contract. And the first step they list, they say, action potential arrives. And it's arriving at the axon terminal. So this cell has been stimulated previously. So imagine this cell, it's stimulated in here at the cell body within the central nervous system. So imagine that stimulus, I'll draw as a little red spike. That's kind of the basic shape of an action potential, what it looks like in the lab. It's able to be propagated along this specialized process called the axon, which travels in one direction this way. So this little action potential, we'll learn how it propagates later, is propagated all the way, to this axon terminal. Okay, so that's step one. The action potential arrives to the axon terminal. Let me, I need some space for to write those steps. I'll add axon terminal. And the way that the motor neuron will communicate with the muscle cell is a chemical interaction. So when that action potential arrives, it triggers this chemical release event. Now what has to happen is that there's these little calcium channels, and those voltage-gated calcium channels open for step 2. So I'll put V-gated for voltage-gated. They open. We're still in the axon terminal. So imagine there's this influx of calcium as a result of the action potential arriving. So I'll put like little in arrows and calcium is a second messenger in some cells. And voltage-gated means a change in voltage kind of opens the gate of those channels. And an action potential arriving would actually do that. And what that does is it triggers this It says calcium entry causes ACH release. So it really triggers this ACH release. What will happen is there are these vesicles of neurotransmitter, and they're just sitting in the motor end plate, waiting to be stimulated to be released, and the action potential arriving, the calcium coming in, it'll trigger a fusion of one of those vesicles to kind of like fuse with the cell membrane, so it's released into this space called the synapse. So imagine, I'll just kind of maybe draw one little vesicle, and maybe it's filled with these little balls, which symbolize the neurotransmitter ACH. Calcium acts as a second messenger to get that vesicle to move to this side of the cell membrane so it can fuse and release ACH by exocytosis. So I'll put 3. Calcium stimulates release of ACH into synapse. You know, the synapse is the physical space between the axon terminal cell membrane and the motor end plate, the sarcolemma. So, I don't know, it's kind of like this space right there. That's the synapse. Sometimes they call it the synaptic cleft. It's a different way of saying the same thing. The physical gap between the two cell membranes. So if you kind of draw like, I don't know, maybe this fuses to that membrane. And then by exocytosis, you release the ACH into the synaptic cleft. And also other terminology that's used, sometimes they refer to this cell of the synapse as the presynaptic cell. It's the first cell that's stimulated, and they call this one the motor end plate, the muscle fiber, the postsynaptic cell. And so this membrane represents the presynaptic membrane, and that represents the postsynaptic membrane. Those terms are used too. I'll just put pre and post, if you ever see those prefixes. Presynaptic cell, postsynaptic cell, presynaptic membrane, postsynaptic membrane, just so you're clear on that. So what's happening here is the first cell did its job. It was stimulated, an action potential arrived here, and it appropriately released its chemical messenger. So the physical communication between the presynaptic and the postsynaptic cell is a chemical one. It's a neurotransmitter called ACH, and that's all the presynaptic cell can do. It depends upon everything here being functional in the postsynaptic cell to work. And so to receive this ACH, my little purplish balls, they diffuse across the synaptic cleft, and they have receptors, which I've drawn here, and they're going to bind its receptor. So that's step 4. Acetylcholine, a neurotransmitter, diffuses across the cleft and binds its receptors in the sarcolemma. I'll paraphrase. ACH binds ACHR. ACH is my shorthand for acetylcholine, and ACHR is acetylcholine receptor. The R is receptor, and sometimes they're called ligand-gated receptors as a more general description. as was the case in a voltage-gated channel, a change in voltage opened the channel for the calcium. But here, a ligand opens the gate. In this case, the ligand is ACH. So you ever see that term, in this example, referring to the acetylcholine receptor. What that does is, when it binds its ligand, ACH, I already drew it open for simplicity's sake. It opens. And what it allows to enter is sodium. Now, they show the exchange for sodium and potassium, sodium going in, potassium going out. I mean, that's fine. I really want you to know that it's the sodium that enters the cell, and that's step 5. The ACH binding opens the ion channel and allows simultaneous passage of sodium into the muscle fiber and potassium out of the muscle fiber. Just know the sodium part going in. All right. Step 5. I'll put sodium influx through the ACHR. Okay, this is all at the neuromuscular junction. So what I haven't drawn yet is this thing called the membrane potential. Before this stimulation event, use a different color. The inside of the cell, of the muscle cell, it was all negative on the inside of the cell membrane. So let me put little negative signs all over the place. It was all negative on the inside of the cell membrane. Here too. What happens is, on the other side of the membrane, there's usually like a positive charge on the outside. And it's like the positive and negative, they're kind of like attracted to each other through the membrane, although they can't actually get together because the membrane is a physical barrier between the two. We call this membrane potential. This is polarized, where it's all negative on one side and positive on the other. It's polarized, that's the term that's used. So imagine all these positives lining up on the outside before the muscle cell is stimulated. But in step 5, we have this reversal of the polarized state, because by these ACHR channels opening, they're going to allow the influx of sodium. So I'm going to draw a bunch of pluses coming into the cell. And what they're going to do is they're going to replace all these negative signs. So now the inside of the cell becomes depolarized. So I'm going to change all the positives, all the negatives to positives, and it's kind of like the reverse is true. Now the inside of the cell has become positive, and the outside of the cell is now negative. So when you have a sodium influx, call that a depolarization. That word is used a lot. It's no longer polarized, you depolarize. You allow positive in. Okay? So what you've got now is a situation where you have all these positive charges that got into the cell, and they look around and they see everywhere else is negative. So positive is attracted to negative, so that's going to move. We call that a current flow on the inside of the cell membrane. I think that's that step isn't mentioned here. On the last step, I should mention that ACH is broken down by ACHE. That's an enzyme that breaks down, and so the ACH can be, it can be recycled and reused. Put that as the last step. ACHE. I'll just draw it in there. It's just going to break the ACH down. So let me write that as a sixth step. I'll leave step 5 there. Step 6, ACHE. E stands for esterase. Let me write the whole thing out. Acetylcholine esterase. It breaks down. ACH. Now that's very important. As long as ACH persists in the synapse, the muscle fiber will continue to contract. So you got to get rid of it immediately so there's not this overstimulation, this unwanted stimulation of the muscle. Okay. So before I erase this last step, that's really the end of it. When you depolarize the postsynaptic cell, you've stimulated the muscle fiber when you've successfully got this depolarization. So is there any questions on depolarization or any of the steps 1 to 5 or 6?
Please explain one time more the difference between polarization and depolarization.
All right. So you have cell membrane, negative on the inside, positive on the outside. Out, in. That's polarized. That's like this very unequal condition of charges. Here's what you would expect in a normal cell. It's as negative or positive on the outside as it is on the inside in this condition. This is not polarized, right? The charge separation is more or less equal. In non-excitable cells, this is usually what you see. Okay, so if you're to like measure this on like a voltmeter, you know, if this is like positive, that's negative. This would read 0. If you could stick an electrode on the inside and on the outside and measure the charge difference, the separation, which is what physiologists do, they would measure 0. But the work of the sodium-potassium pump, it creates this polarized state. So that's polarized. Any question on what polarized means? It's not this. If you're measuring exactly, if you had a little electrode there and a little electrode there, these two locations, going to a voltmeter, what would it read? Positive, negative, or 0 in this condition? Well, I should probably tell you. It would read negative because it always reads with the perspective of the inside of the cell relative to the outside. So it would read negative. Oh, gosh, this should be positive, that should be negative, I'm sorry. So it should read negative. Okay, so any questions about what the difference between, this is not polarized, I didn't write that. It's 0. Students like to use the word equilibrium, right? What does equilibrium mean when I use it to describe this condition? Well, there's no difference between the charge separation outside versus inside. It's as positive here as here. It's as negative here as here. But here, there's clearly a difference. It's negative to the inside. Okay, so that's polarized. So when I say depolarized, when I was doing all this stuff here. So this is depolarized. So what's my volt reader going to mean? Like negative, 0, or positive? Multiple choice. Negative, 0, or positive for this condition? Positive. Positive, yeah. It's going to be like swing back this way. So let's depolarize. Any more questions on that? That's actually really important that you understand what depolarized means. Was there any other questions through 1 through 6? Oh, I erased it. I wrote it up here earlier. Esterase. Basically an enzyme that breaks down ACH because you want to get rid of it. As soon as you get your depolarization event, get rid of ACH or you overstimulate the muscle. That happens like, for example, when weightlifters, like they lift their max, they're just going all out, and there's so much release of the ACH that after they're done to put the weight down, their muscles are still twitching. I don't think I ever lifted weights that heavy, but
This is a little bit more than
get rid of it.
Postsynaptic, how the acetylcholine and neurotransmitter binds to the ligand receptor.
Yeah, just know that it binds to its receptor, so it can open and let sodium in. That's all the detail you need to know. Yeah, I wouldn't know more than that. Well, I don't know more than that. So I'm going to erase this. Let's just look at this picture for a while. So what's happening here before I erase it is they put these little plus charges because they got in here. It was stimulated and all this positive got in. Now, and I said it looks around. It's like, oh, okay, I'm positive. Where can I go? I can go to where it's negative, and that's pretty much, say, anywhere else outside of the motor endplate. This positive influx will travel along the inside of the sarcolemma. So let me write that as a step. So this positive charge here, the sodium influx, sodium ions travel along the inside of the sarcolemma. So this positive charge, it'll just go towards negative, and it'll just travel along. That's what they show you here, okay? You have to look very carefully at the details of these pictures that they give you. Now, I've been looking at them for 15 years, and I've seen everything, but let me show you what you're not seeing. Do you see this right here? Is that positive or negative? You can't see in the back. Those are negative signs. But see that these right next to it? These are positive signs. Okay, so they're trying to show you that it's all negative here, but as these travel, they're traveling to where all the negative signs are, and they're replacing them. Okay, and on the outside it's negative there, and so look at that detail. They're really trying to show you there. Now they're showing you these channels here. All along the surface of the sarcolemma, you have these voltage-gated channels. Right, these are ligand-gated. These are voltage-gated channels. So let's talk about these two. That's for sodium and potassium. So the nerve fiber did its job. You stimulated the muscle, and the action potential is spreading throughout inside the muscle fiber. gated sodium channel. I'm drawing my lipid bilayer here, drawing a little more detail. Let's call this one the voltage-gated sodium channel. Let me draw potassium more. That one's for potassium. So for that one, they're both illustrated right next to each other. They're both embedded in the cell membrane. And voltage gated tells students that, well, what opens and closes these gates that I drew is a change in voltage. So a change in voltage is a change in the charge difference across the cell membrane. And so the difference is this gate, this voltage gated sodium channel has two gates. It has this activation gate and this inactivation gate. Let me just say it has two gates, as opposed to this. I just drew one gate. That's how they draw it. Now, before the cell is stimulated, it's all negative on the inside. And it's all positive on the outside. And this is somewhere else in the cell membrane, that's not a part of the motor endplate. But see what happened, what we just got through diagramming is, you allowed sodium influx at the neuromuscular junction. So you allowed positive in. So let's say it's over here. You allow this sodium influx previously. Right, that's what we got through talking about. And that positive charge gets in, and it looks around, and it wants to travel to where it's more negative, which is pretty much everywhere else. So what will happen is, it'll travel to these negatively charged particles over here. And when ions move through space, I mean, physicists call that current, right? It's local current. Here they call it wave of depolarization. They call it here generation and action and propagation of the action potential depolarized. So these use different languages. I just want you to know that these positives are attracted to these negatives. Okay, here's the thing, other thing you gotta know that I'm not telling you. In this gate, these are all made out of protein. And in this part of the gate right here, it's a little bit positive. Right there and right there. And it's attracted to that negative. So this positive and negative, they're kind of attracted to each other. And they're keeping the gate closed so nothing can get through. Because there's a lot of sodium out here that wants to get in. But it can't because the gate is closed. And it's the same thing over here too. I didn't quite draw it right, but imagine there's positive, negative, and it's keeping the gate closed. So when this positive current comes and replaces these negative signs, replace them with positives. And here too. Replace everything. The inside of the cell became positive, and the outside will become negative when this current just kind of travels by. Now I have a situation where a positive charge is close to another positive charge. So opposites attract, but like charges repel. So that'll swing these two flaps of the gate up to open it. It'll swing it up this way, and it'll swing it up that way. Okay, and that'll open the gate. That's why this is voltage gated. These, it makes this voltage gated channel open, and well, that's how they illustrate it there. They draw the gates open, and they draw sodium going in. Okay, so in your notes somehow, make that gate open. I'm not going to redraw it, but if I were, just the gates would be open, and then sodium would come in. You know, the same thing happens to potassium. They react at the same time. These become positive, and that gate's open too. It just so happens that this gate, the mechanics of it, the kinetics as they call it, are really slow. They react very slow. This one, the kinetics are, they're very fast. So this one opens and lets sodium in. When this one opens, it will let potassium out. But not yet. They respond at the same time, but this one's just much slower. So during the time this gate is open, the potassium gate is still closed. Okay? So now you have to kind of look at the graph that they have. When you measure this in the lab during time, first you're at -70. You're unstimulated. And then when the depolarization comes, you see this spike. On your graph, physiologist in the lab, there's millivolt 0. Here's -70. Here's time. When you're at rest, you're at -70. We call that rest. The inside of the cell is always negative. But when we stimulate the cell, we allow sodium influx. And when this influx comes in and you're measuring it, you can see that. You see it rise from negative, and the influx is so powerful. When you get to 0, which is equilibrium, you don't stop there. The influx is such a mad rush, you actually overshoot 0, and you go all the way . I don't know if you can read that number. That says +30. So the inside is positive relative to the outside. So this side, I'm just going to put dipole. Okay, before I talk about repolarization, are there any questions on the voltage gated channels? How do they work? They're not ligand gated. A change in voltage, the initial influx of sodium here, allowed it to become depolarized, but just in that one location. And so it's up to the, it needs to spread throughout the entire muscle fiber. This is just a very small part of it. Okay, in this slide, what are the authors trying to show you? Well, let me let you read from the slide for a second. Which channel is closed of these two in this picture? They say closed sodium channel. Okay, so what happens is, I said the kinetics are fast. It opens really fast, but it's inactivated really fast. See that ball and chain? That's actually a real thing. It's the second gate, and it has a globular structure. So as soon as that first gate opens, it allows this mad rush in, but then it quickly inactivates. Okay, so that plug right there, they call it sodium inactivation. So let me draw that in my picture. So maybe these gates were open, but then the plug right there, that closes it. Call it sodium channel inactivation. Inactivation. So it's now closed. So what that tells you is sodium's not coming in anymore. But the other gate finally opened. They both started at the same time. Which gate opened first? This one. Okay, for example, if I race a world-class sprinter, 100 meters, the gun goes off and we start at the same time. But who finishes the race first? Not me. Okay. The world-class sprinter does. But I'm like this. I'm like the sodium. I'm like this. The potassium channel here. I finally open. So this gate's finally open, and it allows potassium to rush out. In a previous lecture on the first unit, we said that cells have a lot of potassium inside the cell, so that when a gate opens, potassium will leave out. And we said that cells have a lot of sodium outside the cell, so that a lot of sodium can come in should the opportunity come or arrive. And well, anyways, now you have a positively charged ion leaving. So call this potassium efflux. This gate's open. Potassium efflux, which is the opposite of sodium influx. Something is not coming in, something is now leaving. Efflux. So if a positive charge is leaving the inside of the cell, does that make the inside of the cell more positive or more negative if positive is leaving? Negative. So that'll start to repolarize your membrane potential back to rest. Let me just draw like a down. So we call that repolarization. I'll just put repol. I mean, you're repolarizing, right? Remember how it was before? It was all negative on the inside, and wow. Okay. Are there any questions on repolarization? Yes? Question? Yeah, I'm listening. Don't worry. Take your time.
Repolarization at when calcium and potassium go out outside.
Yes, that is repolarization. Look at the figure. What are these? Positive or negative?
Negative.
Negative. That's what you're repolarizing. Positive leaves, and it allows the membrane to repolarize. And you see that as it going back to rest. Okay. Yeah, so that's what happens throughout the cell membrane of the muscle fiber there. Just depolarization, repolarization, and just to kind of review some of the things here. When we talked about ACH, and you start to study this in your figure, when you look at this figure here, what color are the ACH balls in this figure?
Yellow.
Oh, they look green to me. They're little green balls. Is that what you're looking at there? That's ACH. And I drew them purple in my notes, I know. But they're drawing them green there, and they try to draw them binding their ACH receptors in the motor endplate. But sodium was very important at the motor endplate. Right? So that was another key player that I talked about. Then I talked about ACHE. Think of that as a switch off. ACHE broke down the ACH, so that way you don't get this excessive unwanted contraction. You want to get rid of that in the synaptic cleft. And then I talked about calcium. We're going to talk about that because when the action potential spreads throughout the muscle fiber, calcium will be released so you get cross-bridge cycling. Now there's a few things I didn't list here. For example, in your notes, do you have ACHR? What's the R?
The receptor.
That's the receptor. Don't confuse it with the E. I talked about sodium and calcium, but what about potassium? We talked about it here. Potassium was important for the repolarization. Okay, I didn't put that in that list, but we just know that too. All right, so it's hard to tie this with our case study. The condition is called Myasthenia Gravis, which occurs in about 1 in every 20,000 persons. It's pretty rare. It causes paralysis or muscle fatigue. Pathologically, antibodies attack acetylcholine receptors in patients with MG, as they call it. Therefore, Myasthenia Gravis, by the way, that means muscle, MY, and that just means weak. Myasthenia gravis is an autoimmune disease. We call it autoimmune. Antibodies are normally a good thing, which attack all the foreign invaders in the body. If you get some kind of infection that makes your immune system make an antibody that also recognizes something else in your body you normally want to protect, like ACHR, and that's a bad thing. We call that autoimmune. So in Myasthenia gravis is an autoimmune disease in which patients have developed antibodies that block or destroy their ACHR. So let me write that down. So, in Myasthenia gravis, antibodies attack ACHR. So you have few, many deficient ACHR. So you have fewer effective ACHR. The symptom of this is muscle weakness because the acetylcholine receptors in the postsynaptic neuromuscular junction are destroyed, so you have fewer that are working well. The disease may cause paralysis of respiratory muscles. Okay, if the disease causes paralysis of the respiratory muscles, death can occur, like diaphragm. And that's the main concern. If this happens in muscles that are key to vital organ systems like your lungs, death could occur. Other symptoms may include a generalized fatigue, blurred or double vision because of the eye muscles. Treatment may include thymus removal. They're thinking that that removes the thing that your immune system is making antibodies against. So maybe that will decrease the antibody production that's attacking your ACHR. There's also drugs like neostigmine or some other acetylcholinesterase esterase drug. So what these drugs do, let me give an example here. Treatment RX, neostigmine. What they do is they react with the acetylcholine esterase and thus allow acetylcholine to stay in the synaptic cleft for a longer period. Therefore, the fewer receptors you have that are exposed to ACH for a longer time, so they're more likely to open, maybe even several times, and thus produce a muscle action potential and a contraction. Okay, so it's reason for that. Here's the problem. you have fewer effective AChR. So it's harder to get an action potential in your muscle fiber. You can't depolarize muscle fiber because you have fewer effective AChR. So what this drug does, it attacks ACHE. Do you remember the function of ACHE? It broke down ACH. So what's the effect if you get rid of ACHE? Well, ACH will persist longer in the synapse. Normally that's not desirable. In this case it is, because you have so few of the AChR. If the ACH can persist longer, you allow the fewer ones to open more frequently, and that may achieve depolarization. Okay, so with drug treatment, the fewer AChR open repeatedly to so the cell, I'll say, to reach depolarization. I remember the first time I used this study, I actually had a student who had this. You know, what's the chances of that? 1 in 20,000. So I thought it was a very interesting case to present. And I remember this student, she had to be accommodated. She would tell me her eyes would get really tired while taking a test because it's like, there's this condition. And so I remember thinking, wow, sometimes it's very real for you guys. And she was just like you guys, a young female student, and she was able to get through the class. So the key thing is, you don't want that to affect muscles of inspiration, because if it affects your breathing, that could be lethal. Okay, but things like, well, I can give you a longer time to take the test, or maybe your swim times are slower. That's okay. That's not life-threatening. You can learn to deal with different things in life, but if you can't breathe, that's a main medical concern. I want to move on to this picture here. It's a picture of our muscle fiber, because now that I've taught the neuromuscular junction, I'm hoping this can make a little more sense. What do you think is inside these little yellow balls here? What's inside there? The ACH. Okay, so they draw these vesicles. They even draw them fusing there. I guess if like it's the orange membrane, would you say that's pre or post? Pre. Yeah, pre. The white is post. That's what I would say. Let's move on there. So some more pictures. When you look at the tissue under the scope, you really don't see this detail that the model shows you. So I labeled a few things on this model, which is a different one. This model here, pointing right to there. I just really blew it up. So on 3, that's the postsynaptic membrane. 4, I'm trying to point to the space in between the two. That's the synaptic gap, or you can call it synaptic gap, synapse, synaptic cleft. There's all a different way of saying the same thing. 5 is the presynaptic membrane. 6 is that little white ball, and those are the ones filled with ACH. Okay, 6 and 7, that's a Schwann cell. Those are the cells that myelinate the axon. I took this picture because I wanted you to see the cell surface of the sarcolemma. The action potential travels across the inside surface of the sarcolemma. But in order to penetrate and get to the myofibrils deep inside, you need the T-tubule system. It's this imagination of sarcolemma. And you see how complex it is? It's all colored blue. If I pointed to anything blue here, I'm going for the T-tubule. Remember, T stands for transverse, because it's like trying to get through that section. Remember what we called this stuff here? SR was the abbreviation. Sarcoplasmic reticulum. And what was the chemical that's inside here? Calcium. I think I heard someone whisper that. Calcium. It's important that the nerve impulse, the blue part, reach. I call these end sacs of the SR. They're filled with the calcium. When that calcium is released, all the contractile proteins are here. It will bind up the calcium so you can have muscle contraction. I think that's a good picture there. There's a picture of a motor endplate, or that's the nerve ending, the axon terminal. So let me just call it motor endplate or neuromuscular junction. There's a satellite cell. I didn't mention those. Satellite cells, they have different functions. What they do is they one of their job is to help get rid of, recycle the neurotransmitters. Okay, but it's interesting how these glial cells that help. You remember what the name of this connective tissue covering is? It's a basement membrane with reticular fibers. Endomysium. Yeah, and the cell is inside that connective tissue membrane. So you have endomysium. What's the name of the cell membrane? Sarcolemma. Okay, so they model that sandwiched in between those. All right, so let's get to the crossbridge cycling. I mentioned this before, how, we'll see if you remember. I see 1, 2, 3 things. What did we call it? Triad, yeah. And it's the two end sacs with the T-tubule. Okay, and what they're showing you here is that when the nerve impulse, the action potential, it travels across the surface, when it gets to the T-tubule, it will travel down. And that will allow the end sacs to release calcium. These little channels here, they're sensitive to the action potential there. They're voltage sensitive. And so then that will release the calcium. And, well, I don't know if you can see what color that is. That's a little red ball. And those will allow this here. And we talked about this last time. I don't want to rewrite it on the board. On this top figure, the active sites, covered or not. They are covered, but when it binds the calcium, uncovered, that allows the physical cross bridge to form. So in the next lecture, well, basically there it is again there. I want to go through the steps of what happens when you get that cross bridge formation. So that basically it's attached, cross bridge. You pull, that's the physical work of muscle, then release, and then do it again. You re-energize to do it again. It's a cycle. We'll just go through the cycle one time. And you have to remember what we talked about in the sarcomere. Look at the arrows. These filaments will slide past each other in this direction, right towards the middle. What line is that? M for middle. Okay. Well, anyways, if you just kind of look at what's happening in one thing here. We have all the players. Let's review. Looks like string cheese. The thick filament made of like 300 or something myosins. That's the heavy head there. These little like blue olives, what are those? Actin subunits. And I named all the parts. The rope, the tropomyosin, and the troponin had the 3 subunits. But in this step, let's call it cross-bridge formation. I think your book might call it something else, but this is what I like to call it, cross-bridge cycling. Step 1, cross-bridge formation. Let's note all the events that you should notice in this picture. Calcium has bound troponin. Bounds, binds, troponin. So when that happens, tropomyosin uncovers the active site. active sites. Look like pitted olives. These active sites are exposed. Once they're exposed, the myosin head, it'll bind in this conformation right there. Myosin head binds active site. That is the cross bridge. Okay. The physical interaction of the myosin binding to the actin. Okay. And notice the angle at how it binds. Here's my actin subunit. So anyways, I mean, these angles right there, I mean, they've been measured. I mean, we don't have to know the numbers, but they call this a high energy conformation. It has the ability to kind of like, move that way. They build it as potential to do work. So we'll just call this, high energy conformation. Look at the ATP. There's this binding site here for ATP. You've hydrolyzed the ATP, but you haven't let go of those molecules, the inorganic phosphate and the ADP. They're kind of hanging on. So let me kind of draw that. Kind of hanging on here. PI, ADP. So ATP has been, they say, hydrolyzed. In other words, it's broken down into ADP plus inorganic phosphate, but you haven't let them go. You're kind of holding on to the energy. I'll put that, holding on. I mean, hold on there. The energy is not quite been released yet until you release those subunits there. So that's everything I noticed. Do you have any questions on any of those notes? The next step is the most important step. It's a physical sliding of the movements, and I call it the power stroke. So I'm going to erase cross bridge formation. Power stroke, exclamation, because this is the actual, well, what happens is, let me describe it this way. Step one, the myosin head in the hinge region, myosin head pivots in the hinge region, it kind of moves in that direction. So by doing that, it's pulling the actin past itself. So the myosin pulls actin past the stationary myosin. So that's what a power stroke is. Actin slides past stationary myosin. That head pivots when you release the ADP PI. So it's like you went from that to this. You kind of flexed at this hinge region and you pivoted the head, kind of went like, you know, like flexed there, and this kind of tilted down a little bit. And what you did is you moved from there to there. So you physically moved in this direction. Well, this will be towards the M line. You're moving towards the M line. So this is the working stroke. It's the physical moving. This is why they call it the sliding filament theory, because actin is actually sliding. All right, any questions on that? That's it, but that's muscle contracting, okay? And if you look at our figure, they draw both arrows, you know, going in. You know, you're shrinking that H zone, as we talked about the last time. Now you have to kind of like get ready for the next one. So what happens is the first thing you have to do is detach and kind of like reconfigurize yourself. So in the next step, ATP is necessary. ATP is necessary. Because on the previous step, you let those guys go. So let me put that as the last note here before I move on. Like ADP and PI, they're gone. You've released them. So you kind of like need ATP again to start things over. So in this next step, cross-bridge detachment, ATP rebinds, that allows this cross-bridge detachment. Notice how calcium is still there. Okay, so as long as the active sites are there, you know, calcium, if calcium's there, the active sites are exposed. So you need ATP to kind of like detach. I'm just writing ATP required for this detachment. So let me kind of draw it again. So yeah. So ATP is attached there, and it's, let me draw the actin. It's detached. They call this a low energy configuration. That myosin head is still like kind of flexed forward and it can't really do any work in this configuration. So ATP has caused this detachment, but it's called low energy configuration. In the last step, the myosin head re-energizes. So what happens is you hydrolyze it but hold on to it. So basically I'm going to draw a big arc going from there back to there. You go back to your high energy configuration. Okay. But this is just prior to cross bridge formation. Okay, you haven't formed it yet, but you will. As soon as you recog, you're going to attach. Myosin head pivots back. Sometimes they call that it recocks to a high energy configuration. As long as calcium is present, this will cycle again and again and again and again until you get the force you need from your muscle. If you're done, you needed ATP to, well, let me write this down here. I'm kind of done talking about the cross-bridge cycling. Know why you needed ATP. You needed ATP is required for two reasons. Number one, you needed ATP to break the cross bridge, okay? You also need ATP to pump calcium back into the sarcoplasmic reticulum, get rid of it. Pump calcium back into SR. So that way the muscle contraction can end, depending on when you want it to end. I mean, if it's, as long as calcium is there, this cycle will continue. Okay. As long as ACH is there, the muscle fiber is stimulated and calcium is released, and calcium will be there. And so you have to think all the way back to the neuromuscular junction. How do you get rid of ACH? What's the molecule? ACHE. Once that's gone, then ATP will pump the calcium back, and then this can stop. But if it's calcium is still there, you just do it again. Let's take a break. I'll come back at 9 o'clock. We'll have to continue on with this lecture.